Hello everybody, today I'm going to talk about muscle contraction. I hope that you will enjoy this lesson. We have three types of muscle in our body. So we have skeletal muscles, which are voluntary muscle or striated muscle. What are voluntary? Voluntary muscles, that means I control my movement. I could control my movement, so I could decide to open my eyes or run or walk. So I take the decision to do this action. This is voluntary. We have here cardiac muscle, which is involuntary, because here an example of cardiac muscle is our heart. So we can't control the beating of our heart. Okay, this is involuntary. And here the third type here is a smooth muscle, like our stomach, our urinary bladder, or the uterus. So here they are also involuntary, so we can't control their movement. So now I would like to talk in more details about skeletal muscle, because I am talking about muscle contraction. So skeletal muscles, we have approximately 650 muscles attached to our bone. So here we have the skeletal muscle, which is attached to our skeletal system, so they name it skeletal muscle. And here we also could name it voluntary muscle, because we control the movement of our muscle here. So when I decide to open my eyes, that means here there are signals going to my brain, and these signals will pass through neurons, and then will be going to the muscle and ask this muscle to contract. Okay, so then the muscle will contract according to my decision. And here we could name it as a striated muscle according to the unit of the muscle contraction, which is the sarcomere. As a sarcomere here, you see it consists of here. It is the board is the Z line, and it has a striated muscle here. Why? Because we have actin filament, which is in green, which is a thin filament, and we have myosin filament, which is in purple, which is a thick filament. The actin and myosin filament, they are arranged in a striated form, so we could name it a striated muscle. The name striated muscle comes from here. And here is the sarcomere, which is the unit of muscle contraction. So each muscle, our skeletal muscle, contains of bundles of muscle fiber, and each bundle of muscle fibers contain myofibril, and each myofibril contains sarcomere. Okay, so here is a picture which demonstrates this lady. She has her muscle here, and this muscle consists here a bundle of muscle fibers, and these myofibrils, they have, as you see here, nucleus. Okay, they have many nucleus on it, and they have numerous myofibrils here, and here is an extension of this myofibril, which consists of sarcomere here. And here you could see the sarcomere has an M line in the middle, and the board of each sarcomere is a Z line. Okay, and here is a thin filament, which is actin, and here is a thick filament, which is myosin. And this is an electron microscope of this muscle. You could recognize here when the M band here in the middle, this is the M band. Okay, and here we have H zone, which demonstrate only myosin filament. Okay, and here is the I band, which present only actin filament here. And here we have overlap of both actin and myosin, which gives us this dark area. This is the A band, where there is overlapping of both actin and myosin together. Okay, and this is the whole sarcomere, which is the unit of our muscle contraction. So here, this is a sliding filament mechanism. So I would like you to know first some scientific terms like ATP. What is ATP? It is adenosine triphosphate, which is the source of energy in our body. And when it is hydrolyzed, it gives ADP and phosphorus. Okay, we have action potential, which happens in the nerve cell when there is disturbance in the membrane. That happens when we receive any external stimuli. So this disturbance changes the charge within the cell, or there is a disturbance in movement of ions. So it creates a positive energy inside of the neuron, and outside become more negative. Okay, and this pass through the nerve in only one direction. I talked about it in my previous lesson. We have also sarcoplasmic reticulum. What is sarcoplasmic reticulum? It is the endoplasmic reticulum within the muscle, and this is a place where calcium is stored. Okay, so this is very important for muscle contraction. And we have acetylcholine, which is a neurotransmitter released from the end of each neuron, because they have synaptic vesicles, and this synaptic vesicle, when they received an action potential, then they are released and they go to the surface of the receptor, which are located on the muscle or the sarcolemma, and then they transmit another action potential to stimulate the release of calcium from the sarcoplasmic reticulum. So here we will see that we have here this is a contracted sarcomere, and this is a relaxed sarcomere. So you could find that the contracted sarcomere is shorter than the relaxed sarcomere. Okay, and here we could talk about the sliding filament. We could see here we have, this is actin filament, which is a thin filament. It has a tropomyosin, which is a thin filament. Okay, and it has the troponin here, and when calcium is released, it is attached to the troponin, and then it pulls the tropomyosin away, which will cause exposing. It exposes this myosin binding site. They will be exposed. They will be exposed to be attached to this head, the myosin head. Okay, so this is the thin filament. Here it is a thin filament. So when we start this mechanism of contraction, the sliding filament mechanism, we will recognize that the ATP here is detached, and to give ADP, and here it is hydrolyzed to give ADP and phosphorus. Then the head of the myosin is attached to the myosin binding site through a cross bridging, and then after releasing of adenosine diphosphate and phosphorus, then the head move inward here towards the M band, so it enhances the contraction of the sarcomere and becomes shortened. Okay, and then another ATP is attached. So when ATP is attached, there is detaching of the head from the actin filament, and then another ATP is hydrolyzed to give ADP and phosphorus, and the process is repeated again and again until the muscle is contracted to give this short sarcomere. So here is a summary. We could recognize that here is the muscle fiber, which contains, here this is the sarcolemma, and each muscle fiber contains many nucleus. And here under the sarcolemma, which is the membrane of the muscle, we have sarcoplasmic reticulum. This is in blue, and the violet are the tubule. Here they magnify this part. Okay, so you could find this is a violet, is a tubule, and here is a sarcoplasmic reticulum. And those are many myofibrils, which here, when they are extended, each myofibril consists of a unit of muscle contraction, which is sarcomere. So here we could see that when a motor neuron receives any action potential, it passes it through the nerve and through the muscle. And here is a nerve. When the action potential passes here, it goes to the synaptic vesicle and causes the release of acetylcholine. When acetylcholine is released, then it is attached to the membrane, the receptor of the sarcolemma here on the membrane of the muscle. Then this action potential passes down through the T-tubule to reach the sarcoplasmic reticulum, and then it stimulates the release of calcium from the sarcoplasmic reticulum. So when calcium is released, it is attached here to troponin, and when troponin is then it pulls away the tropomyosin, so in turn the myosin binding head got exposed. They got exposed to the myosin head here. And when ATP is hydrolyzed to give ADP and phosphorus, the process of contraction happens by moving this actin filament inward towards the M line. And here is the relaxed form of the muscle, and here is the contracted form of the muscle. Here is how to communicate with me. You could communicate with me through my Twitter account or through WordPress or through this channel by leaving any comment or asking any questions. And here are the references that I use through this presentation. And here are the questions that I would like you to answer, please. So you could communicate with me through the previous links. And thank you very much. And I would like you to watch a certain video on YouTube, which is a 3D video related to muscle contraction. And I hope that you will enjoy.
You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction, or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross-bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
Thank you very much for watching, and I hope that you enjoyed this video.